Wanna dance by the water neath the Mexican sky. Drink some margaritas by a string of blue lights. Listen to the mariachi play at midnight. Are you with me? Are you with me?
I'm very happy, and this it's a real pleasure to introduce Professor Rosa Lozano-Durán, who is an amazing scientist and also a very good friend. So I'm truly happy to have the opportunity to have you here. So Rosa is full professor at the ZMBP in Germany. She studied biology here at the University of Malaga and also completed her PhD here under the supervision of Dr. Eduardo Bejarano, working on Gemini viruses. In 2011, she moved to the Sainsbury Lab in the United Kingdom for her postdoc, where she stayed in the plant pathogen field, but in this case, working with bacteria. Right. So she spent 4 years there, working first with Silke Robatzek and then with Cyril Zipfel. And in 2015, she moved to the Shanghai Center for Plant-Based Biology in China to start her independent career. There, she went back to her beloved Gemini viruses, and she started and consolidated the Gemini team lab there. I actually think that this moving to China to start her first independent experience shows a lot about Rosa, how strong she is, her bravery, how she pursued her passions. And she took advantage of the conditions there in China to really make and create a great lab. In 2021, she came back to Germany came back to Europe with her group, when she got her position at the ZMBP. Both in China and in Germany, she combined her outstanding science with teaching and institutional activities. She is now managing a lab, directing a lab composed of 10 postdocs, 7 PhD students, 2 technicians, not counting all the master students and undergrad that are there. So it's truly amazing. She has authored more than 100 papers, and she has signed as corresponding authors in journals as important as Cell, PNAS, Molecular Plant, EMBO Journal, and also has written several annual reviews. And also has got funding from the major funding agencies, so she has an ERC Consolidator grant, and also won the €1 million grant from the Volkswagen Foundation, so it's truly amazing. Also, her career has been recognized by several awards. Her list of awards is truly impressive. The latest one has been that she's been recognized EMBO member now this year in 2025. Beyond this, all of us who know Rosa know that she's a very passionate, positive, uplifting person. Her enthusiasm is really contagious. She's a really persona vitamina. And this, combined with her excellent mentoring skills, makes that everywhere that she goes, she creates a very special environment around her. And on top of that, she's an amazing friend, a caring one, and that she always will be there for you. So thank you so much for accepting my invitation. Do your magic and hypnotize us.
Thanks a lot, Karen, for the amazing introduction. I asked you not to make me cry, and you were very close. So it's really a pleasure to be here. Thanks very much for the invitation. It's very special for me, because as Karen said, I actually studied here, and I did my PhD in what already on paper was IHSM back in the day. So many of you know this. What you might not know is that actually I was destined to be a marine biologist. So here you can see me taking samples from my research at a very early age. These included tasting experiments. I was very committed. And shortly after these pictures were taken, I actually published my first paper, which was on echinoderms as bioindicators of the quality of the waters in our coastal area. So as you can see, I was really on track. So what happened then, you might wonder? Well, my future as a marine biologist was snatched from the claws of destiny by my second-year genetics professor. And he was so inspiring intellectually that I had to follow him. So you can see me here during my PhD, all happy to have wasted all those preliminary data that I had already collected. You might recognize some other familiar faces there on that picture. We were a little bit less experienced at the time. So this is just to say that I could be talking to you today about dolphins, but instead of that, it's going to be plants. Well, so plants. I think everybody in this room is aware of the fact that plants, as opposed to animals, have constant development. And this means that they will keep producing new organs throughout their lifespan. And while the growth of these organs is quite plastic, their form and function is actually remarkably robust. So if you take a leaf, for example, that's a flat photosynthetic structure. So a leaf will develop as a leaf regardless of temperature or water regime. A leaf will be a leaf, almost always. So under certain circumstances, these papaya leaves here, which should be flat like the ones on the left, become these crumpled and almost unrecognizable structures. Now, what is the force that can override a developmental program that can withstand heat and drought and flooding and cold? So the answer is just a tiny piece of extra DNA. That's the only thing this plant here has that this one doesn't. Of course, because you have seen the title of my talk and you have heard Karen's introduction, you can make an educated guess, and you'll be right to say that this piece of DNA is probably a virus. So it's more specifically a Gemini virus, and I know many of you in this room are familiar with this family of viruses. They infect more than 600 plant species worldwide. Disease outbreaks can lead to complete crop losses, and currently we don't have direct control measures. The very scarce genetic resistance that is available is frequently overcome because these viruses evolve incredibly fast. So Gemini viruses are a real-world problem. But in addition, they are also intellectually very appealing. So here again, you can see an infected plant, in this case a tomato plant, as opposed to an uninfected healthy plant. And you can see how symptoms are developing here. So the plant is clearly smaller, the upper leaves are curling, and they are yellowing in color. And that is because the virus is rewiring plant development and physiology, which I find remarkably per se. But I hope you'll agree with me in that this is even more striking if we take into account genome sizes. Because while this plant being infected here has a genome of roughly 900 megabases, the virus has a genome that, as I mentioned, is 3 kb. So that's smaller than your average cloning plasmid. I could say this a little bit differently. If the plant genome was to stretch across 3 football fields, the virus genome would be the size of a grain of sand. And yet, I think it's pretty clear who has the upper hand in this interaction. So, in a nutshell, what we are trying to understand in our lab is, how is this possible? How can this tiny, tiny biological entity, which is not even alive, take control over a much more complex, multicellular eukaryotic organism? So, we are working towards understanding how Gemini viruses cause disease. So what we know is that, in short, these viruses are transmitted by insect vectors. Then once the virus is in the plant cell, the genome has to be released. It has to enter the nucleus where replication will ensue. Then the virus also has to move. This encompasses intracellular, intercellular, and long-distance movement. And of course, it has to suppress the plethora of antiviral defense responses that the plant is capable of deploying upon perception of the viral threat. Now, if the virus manages to tick all of these boxes, then we will have a systemic infection and probably symptom development. But if you would like to be with the underdog and you are more interested in the plant than in the virus, then I will tell you that these viruses are also excellent probes to understand plant processes. And this is precisely because the virus has to tick all of these boxes. So these viruses can help us understand processes as fundamental as cell cycle regulation, DNA replication, epigenetic regulation, transcription, splicing, translation, you name it. And I would argue that Gemini viruses can very well be the plant SV40, and as SV40 did for animal eukaryotic biology, really illuminate our understanding of plant biology. So the genome of a Gemini virus looks roughly like this. So this is the genome of what has been so far our favorite Gemini virus species, tomato yellow leaf curl virus, or TYLCV. It's just 2.8 kilobases in size, and it contains bidirectional and partially overlapping open reading frames that you can see here depicted by arrows. Now, this circular single-stranded DNA molecule is encapsulated in this beautiful geminate particle that gives the family the name. So for the past 10 years now, we have been, as I said, trying to understand how these viruses really work. And we have found a number of things. For example, we have found that these viruses tend to target hub host proteins and therefore have an outsized effect by protein-protein interactions. We have also seen that these viral proteins are multifunctional. Of course, this was known before. We just have a few more examples now. They repurpose the host molecular machinery for viral functions, and they can have combinatorial functions. So basically, when you put two of these proteins together, they can acquire novel activities. And finally, these viruses seem to have evolved strategies to maximize their coding capacity that we were not fully aware of before. And today I'm going to touch upon three of these points here. So one real challenge when studying Gemini virus infection is the fact that for many of these viruses, there is a very clear tissue tropism. So they are restricted to phloem companion cells. So that's what you can see here. This is a tomato plant. This is a cross-section of the stem, transversal section of the stem, and a cross-section of a leaf. And this plant is infected. And the cells that you can see in magenta are the ones where the virus is replicating. So you can very clearly see that this is really a minority. So it's just one cell type. And this actually implies that when we do any kind of bulk analysis like RNA-seq, there will be a massive dilution issue. So it's very difficult to reach conclusions. So to try and overcome this limitation, Tan, a very brave postdoc in my lab, decided to try a single-cell RNA sequencing. So he used tomato plants for this. He spent 1 year setting up the protoplasting so that he could effectively isolate phloem companion cells, and then he just gave it a try. And all his hard work really paid off, because at first attempt, he got a beautiful cell population. So when he did the UMAP clustering, he found all main cell types that you would expect in a tomato leaf. And something interesting that I will come back to later is the fact that when he checked the percentage of cells that he found in his sample, he saw, when comparing the infected to the uninfected plant, that there is a very clear increase in vascular cells. But how good are our data? So how much can we really learn about the infection? So to try to see how complete our data set was, we looked at the viral transcripts. So in the viral genome, we have these genes here on the complementary strand. These are early genes, so they are expressed early during infection. And these ones here are late genes. So when we checked where we could find the viral transcripts, we saw that they were all in this region here. That is the vasculature, and more specifically companion cells. And when we cluster these cells, we can find some expressing C1 alone. That's probably a very early stage of the infection. And then early genes alone, all of the genes, or late genes alone. And this makes us think that we are indeed capturing the complete infection cycle. So we were very intrigued by this observation that there's more vasculature in infected plants. So Tan decided to check whether this is something that we could actually see. So here you can see the vasculature in a cross-section of a petiole or a stem. And this is a time course of the infection. You have the control samples on the top and the infected samples here. And I think you can already distinguish how, indeed, we can see an increase in vasculature in the infected plants that can be observed already at 14 dpi and is very clear at 21 dpi. Just in case you don't trust an image, he also very patiently did the quantifications. So you can see the cell numbers here and the vascular ratio here, and again at these 3 time points. And we can very clearly observe how there is a significant increase both in cells numbers and in vascular ratio. So indeed, the viral infection is triggering apparently an overproliferation of the phloem. Now, is this actually relevant for the infection, or is it just an unintended byproduct? So to try and gain some further insight into this, we used 2 known phloem regulators. So one of them is a UL, so this is a negative regulator of the phloem because it negatively regulates SMAXL4, which is a positive regulator of the phloem. And the other one is SMAXL4 itself. So if we silence UL, we will have more phloem. If we silence SMAXL4, we will have less phloem. So TAN silence these 2 regulators in Ecotiara benthamiana. You have the pictures of the silence and infected plants here on top at 2 time points, early and late. You can see that both genes are very efficiently silenced, and then we look at viral accumulation, and we can't find that when we have more phloem cells, indeed we have an increased viral titer as opposed to what we have when we have fewer phloem cells here. And this pretty nicely correlates with symptoms as well, which are stronger in the UL silence plants and milder in the SMAXL4 silence plants. So indeed the virus seems to potentially increase the number of phloem cells manipulated in development in order to create more niche where to replicate and accumulate. But the nice thing about having this data set now is that we can ask all kinds of questions. So what you see here is a reclustering only of vasculature, so we can get further insight. And in this case, we see all the cell types that are present in the vasculature, including here companion cells as cluster number 6. And here, this is a cluster that we have called responsive cells. Because if we look at what happens in the control, this cluster here is simply not present, right? But it is present in infected samples. And it is indeed responding to the infection. So we can ask, what is the main defensive response that the plant is actually presenting to the viral infection? So we took these responsive cells and we did functional enrichment analysis. And then the first category that we find, which is very highly enriched, is regulation of salicylic acid by a synthetic process. So the plant is producing salicylic acid in response to the virus. We can also find further support for this if we look at the expression of SARD-1. So this is one of 2 main transcription factors regulating the expression of the SA biosynthetic genes. And this expression coincides with these responsive cells, and there is a gradient, as has been suggested in different works for SA responses in response to pathogens before. And this actually is also supported by some experimental data that we got some time ago, where we saw that this is an infection in tomato plants, these are Naji tomato plants, so these plants express this transgene that actually depletes SA, so these plants don't accumulate SA as compared to the wild type, and you see how the virus accumulates much more in the Naji plant. So indeed, salicylic acid seems to play a very prominent role in the defense against Gemini viruses. Which, of course, brings me to my next question, which is how do these viruses overcome the plant defense that the host is actually deploying? And this is a perfect segue for me to now tell you a little bit more about one of the viral proteins that we have been working on for some time now, which is C4. So C4 was until recently considered the smallest protein encoded by Gemini viruses. So it's 97 amino acids in size, and it contains 2 targeting signals. There's a meristylation site in position 2. So meristylation is a lipidation that tethers proteins to membranes. And then there's a chloroplast transit peptide that spans roughly half of the protein and would target the protein to this organelle. So as you can probably notice, these 2 targeting signals are mutually exclusive because the protein cannot be at the plasma membrane and in chloroplasts. So we were very curious to see what would happen to this protein when expressed in the cell. So we found that upon expression, C4 is meristoylated, probably because this modification is co-translational, and then it goes to the plasma membrane. So that's what you can see here. That's C4-GFP associated to the plasma membrane. And we found that at the plasma membrane, C4 interacts with some receptor-like kinases, including one called BAM1 and its closest homologue BAM2. This interaction happens particularly strongly at plasmodesmata, as you can see in this biopsy experiment. And what we found in one of the first papers from the lab, of which the first author is here in the audience, BAM1 and BAM2 are positive regulators of the cell-to-cell movement of RNA silencing. And through this interaction, C4 is suppressing this function. But this became even more interesting when we realized that when defense is activated, and this is something that happens to some extent when the virus is infecting the cell, then C4 relocalizes from the plasma membrane to chloroplasts. And that's what you can see here. So this cell here is expressing the same protein as this one, C4-GFP, and the only difference is this one is now infected. So you can observe how C4 is being depleted from the plasma membrane and it starts accumulating in chloroplasts there. So in the chloroplast, C4 interacts with another plant protein that is called Cas for calcium sensing receptor, and probably through this interaction, it suppresses the downstream activation of salicylic acid signaling. So C4 has two distinct roles in two different subcellular compartments, and in both cases, of course, in the end, helps to promote the viral infection. So we think this is a really nice example of how subcellular compartmentalization can underlie multifunctionality of a viral protein. And interestingly, we also found that this connection between plasma membrane and chloroplasts is not something that TYLCV has come up with. So indeed, this pathway seems to exist already in plants. We have found at least one protein, CPK16, which is a positive regulator of defense that follows this pathway as well. And from the chloroplast, in this case, CPK16 activates defense. And then we found that different pathogens, including viruses and bacteria, seem to have convergently evolved to hijack this pathway and send effectors to the chloroplast from where they actually suppress defense. So this is a very nice model, if you ask me, but it still leaves some open questions, particularly pertaining to this area here. Namely, what is the trigger that leads to the relocalization of C4? And how does it actually physically move? So the first question is something that our famous Laura is trying to tackle. And the first thing that she did, because we knew that infection led to relocalization of C4, was to actually test each of the viral proteins independently. And he found that expressing Rep was sufficient to trigger this relocalization. So we were very happy because we had narrowed down this effect to only one viral protein. But we were also a little bit puzzled because there was a very clear spatial discordance. Because Rep is a nuclear protein. So how can a nuclear protein trigger the movement of another protein that is at the plasma membrane? So we were banging our heads against the wall for some time until we realized that if we looked carefully, we could find another open reading frame that was included in that of Rep in the opposite direction. And this open reading frame, which we call PVG, for peptide encoded in the viral genome, produced a peptide with this sequence, so rather short, but that had a predicted secretion signal. So then the mature peptide would be like this, and this mature peptide would be in the apoplast. And that is indeed what Laura saw when expressing this PVG gene fused to M. cherry and then did plasmolysis. So you can see the red signal there, actually outside of the cell. So we reasoned this was much more plausible, that the virus produces this a little peptide that is secreted and from the apoplast then triggers relocalization of C4. So she did some sanity check first. So she saw that when she expressed this PVG alone, then she could see some degree of relocalization. And on the contrary, if she mutated this new open reading frame in the Rep open reading frame, then C4 did not move anymore. So it was indeed not Rep. It was this tiny PVG inside of Rep. So to confirm that it is indeed acting from the apoplast, she extracted apoplastic fluid from different samples, some mock samples, some transformed with an empty vector, and some infected with TYLCV or expressing the rep. And only in the case where the virus or the rep with the PBG were there, then she could see relocalization of C4. So indeed, this is happening from the apoplast. But of course, this would imply that this peptide is recognized by the plant somehow. So we synthesized this predicted secreted peptide, and we treated Nicotiana benthamiana plants with it, and then we did RNA sequencing analysis. And we found that there were more than 4,500 genes that were differentially expressed. So definitely the plant is sensing this peptide and is responding to it. If we do functional enrichment analysis of the upregulated subset of genes, then we find geo categories that are overrepresented that are linked to abiotic stress or to defense. So this PBG in the apoplast is perceived and leads to an activation of defense. But is this something that is good for the virus? So does this peptide have a pro-viral effect? So to test this, we generated a mutant that can no longer produce this PBG, but otherwise is not affected in anything, and we infected tomato plants with it. And we could find that indeed there was a very clear penalty. So PBG has a pro-viral effect. Then we reasoned maybe what PBG is doing, because remember that C4 moves when defense is activated. So maybe PBG is activating the bare minimum amount of defense that you need to move C4. And if this were the case, then we should be able to complement this by targeting C4 to the chloroplast independently. So we generated some transgenic tomato plants expressing a non-myristoylable version of C4. So we can actually mutate the glycine in position 2, that is the residue that gets myristoylated, and then the protein can no longer be modified, and it goes to the chloroplast directly. So when we inoculated these plants, and you can see the results with 2 independent lines here, we found full complementation of the PBG mutant, suggesting that indeed this triggering the relocalization of C4 is the main function of this peptide. So what we think is happening here is that PBG is acting as a viral mimic of a pathogen-associated molecular pattern. So it's not triggering a full-blown PTI response, but it's triggering a sufficient response to trigger the relocalization of C4 and therefore mediate a timely suppression of defense. And I will be very happy to discuss this further later if you're interested. Now, the second question, how does C4 move, or CPK16 for that matter, from the plasma membrane to chloroplasts, is what fantastic postdoc Ji Hao is trying to tackle. So what he has found, to make a long story short, is that C4 and CPK16, both of them, get an endocyte host when defense is activated. For example, when he treats the plants with the bacterial peptide FLAG22. And you can see the colocalization of these fluorescent proteins with FM464, which is a membrane dye, or here with an endosome marker that is ARA7. And if he impairs endocytosis, for example, by expressing a dominant negative clathrin heavy chain, then C4 is retained at the plasma membrane, and this is something that he can actually quantify. So it seems that there is an endocytic pathway that links plasma membrane to chloroplasts, and that is activated upon pathogen perception. And this works both for C4 and for CPK16. But this leads us to another question. What triggers this endocytosis? So when do the proteins know when they have to go in? And for many plasma membrane-associated proteins, the trigger is actually ubiquitination. Interestingly, by mass spectrometry analysis, we found ubiquitination in C4, and one of them was in this lysine here, in this little sequence here. And some of you in this room, at least 1 or 2 people for sure, will recognize this sequence, because that's what we called before the CCL-like domain. Interestingly, when we look at CPK16, there's also a CCL-like domain. And because we worked on this before in a different context, we already had 3 mutants of this domain in C4. So Ji Hao tested ubiquitination of these proteins. So he has the wild type here, the 3 mutants, and a fourth mutant that lacks the glycine in position 2 and therefore doesn't get myristylated and goes to the chloroplast. And then you can very clearly see how, despite a similar accumulation of the protein, we can detect polyubiquitination of the wild type protein, but not the mutants, any of the mutants, not the mutants in the CCL-like domain or the one that goes to chloroplasts. So C4 seems to get ubiquitinated at the plasma membrane, and this seems to depend on an intact CCL-like domain. And if Jihao treats his samples with a ubiquitin inhibitor, then again C4 gets retained at the plasma membrane. This is something that he can quantify, and the mutants do not get internalized as efficiently. So there's a very clear delay. So it seems that ubiquitination is indeed required for the endocytosis of C4. But if this is the case, then what is the E3 ligase that would be doing the job? So because Jihao is not easily scared, he decided to look for it. And for this, he used turbo ID-based proximity labeling experiments. So he took C4 and CPK16 and all the controls, and then he compared everything so that in the overlap, he would only find whatever interacts with the protein that moves. Nothing that is at the plasma membrane, nothing that is in chloroplasts, only whatever interacts with the proteins that are translocating. And in this 152 protein set, he found only one E3 ligase. And that one E3 ligase is RGLG2. So in Benthamiana, we have 4 members in this family, and all of them, as you can see when we compare our RNA-seq data from uninfected or infected plants, all of them are induced by the presence of the virus. And if he silences the members of this family, then again, C4 gets retained at the plasma membrane. And I don't have time to show you this today, but we also have in vitro ubiquitination data showing that indeed C4 and CPK16 are specific substrates of RGLG2. So this is the model that we are currently working on. So we think CPK16 and C4 are sitting at the plasma membrane until, upon a biotic threat, RGLG2 is somehow activated, and then it ubiquitinates these proteins that then enter the endocytic pathway. And Jihao has spent quite some time dissecting this pathway, but in the end, these vesicles cluster around the chloroplast, and again, this is what we see in this image that I showed you earlier. So this clustering of vesicles around chloroplast, this is something that we can only observe when we activate defense. We think then these proteins are delivered inside the chloroplast, and from the organelle they can regulate salicylic acid-mediated defenses. And I just wanted to mention that C4 does some more things. One of them is to actually induce symptoms. So this is work that we published earlier this year, and again, some of you might recognize it because Emmanuel is co-first author, and he's in the audience, and the work has been done in collaboration with Eduardo and Juan Antonio. And before I move on, I would just like to show you how all of these functional information is actually distributed in this tiny protein. So as I mentioned, C4 is 97 amino acids in size. There's this myristylation site in position 2. Myristylation is essential for the localization of the plasma membrane, so therefore it's essential for C4 to suppress the movement of RNA silencing, but also to produce symptoms. Then we have the chloroplast transit peptide. It's already overlapping. And this part of the protein is essential for chloroplast targeting, so it's required for the suppression of SA-mediated defenses. But interestingly, this is also the part of the protein that interacts with PAM1, with the receptor kinase. So it's also required for the suppression of silencing movement. Then we have there the CCL-like domain, which is essential for the interaction that leads to symptom development. But as I just showed you, it also seems to be essential for the relocalization to the chloroplast, and therefore it's also required for the suppression of SA defenses. And then this rest of the protein, this is the mature protein that will be in the chloroplast, and it's therefore the part of the protein that interacts with this Cas protein, and again leads to the suppression of SA. So as you can see, the information is super compact and everything is overlapping. So if you are not impressed by this still, I will remind you that C4 is encoded by this gene over here that is fully embedded into the bigger C1 or rep gene, encoding the replication-related protein, which really does not have a lot of leeway for changes. So how all of this evolves at the same time, that's something that I'll probably never understand. Okay, so I have told you C4 is multifunctional, and I hope you are convinced of that by now. But still, if we take one step back and we look at this again, I can't help but wonder something that I have been asking myself for the past 10 plus years. Like, really? It doesn't matter how multifunctional they are. 6 proteins, is it sufficient for this? And actually the answer is no. So if we look again at the T-LCV genome, we said early genes here, late genes here, right? So if this is all there is, if we now do long-read RNA sequencing, we should find reads in one of the strands here and in the other strand here, right? However, that's definitely not what we find. So when we do long-read RNA sequencing, we find reads spanning the entire genome on both strands, including reads, for example, in this region that is supposed to be non-coding. Now, this is not something that is specific for T-LCV. We have also done the same experiment with 2 more Gemini viruses belonging to different genera, so a Curtovirus, VCTV, and a Mastra virus, CPCTV, and in all cases we see a similar scenario. And some of these reads that are not where we would expect them to be support open reading frames that we can actually predict. So we are pretty convinced that we do have novel viral genes. Also, there are novel splicing-derived protein isoforms. I don't have time to tell you about that today, but wonderful postdoc Delphine, hopefully soon wonderful PI Delphine, I think told you about this recently. So we are trying to test whether these new genes are really leading to proteins. So we are testing whether we can detect the proteins during infection by doing IPMS and also ribosome profiling in collaboration with Karen here. We are testing whether the putative proteins are relevant for infection by doing mutant analysis. And if the answer to these questions is yes, then we are trying to understand what the protein function is. So in brief, we are finding proteins that show novel subcellular localizations that have not been seen before for Gemini viral proteins. We find convergent in targeting of organelles such as mitochondria and also novel functions. And at the moment, considering the experiments that we have done so far, we have already moved from 6 proteins in T-LCV to at least 25. So I would just like to give you a flavor of the kind of thing that we are finding. So first I would like to say that we can find new genes that help with virulence or actually help with resistance. So these are results on CPCTV that have been generated by a recent very valuable acquisition, Axel. So these are infected Benthamiana plants. These are the ones infected. This is one infected with a wild-type virus, so with the typical symptoms. This is a mutant that leads clearly to milder symptoms, and this is a mutant that leads to heightened defense. And in both cases, symptoms correlate with differences in viral accumulation. And I would like to give you one example of CPCTV, and this is work by a former postdoc Yu, who is now a PI at the University of Shenzhen. So she was working on CPCTV, which is a little bit different from what I showed you before because it's a different genus, and it has relevance because it leads to up to 95% yield losses in chickpea. And when she was looking for putative new genes, and here she was comparing 41 species, and she found the canonical proteins. When you compare the number of hits with the protein size, the canonical proteins appear here. The one I'm interested in right now is the one encoded by that gene here. We call it ORF57, and even though it's not very highly represented in the subset of viral species, it's present in 100% of the dicot-infecting Mastra viruses. So I have to say Mastra viruses mostly infect monocots, some of them infect dicots, and all of them seem to have this ORF57. And importantly, we have already identified the protein in mass spectrometry analysis from infected samples, so we are sure the protein exists and is there. So when we use AlphaFold multimer, we find that ORF57 is predicted to form this beautiful channel-like structure, which is possibly a hexamer. We can detect the monomeric interaction between different subunits of ORF57. I'm showing you here the result from split luciferase assays, but we also have co-IPs and yeast hybrid. And because we can predict the interaction, we can actually generate a mutant that loses the interaction. So we modify 2 residues, and you can see the mutant here loses the capacity to self-interact. And importantly, if we use blue native page, then we can see the formation of a higher order complex when we use the wild type protein, but not with the mutant protein, even though in both cases the protein is equally accumulated. So indeed, this new viral protein seems to form a complex. And this protein is required for infectivity, and complex formation seems to be essential, because if we infect Nicotiana benthamiana with a mutant that no longer produces ORF57, then it's really impaired in infectivity. So this is viral accumulation here. And if we use a mutant that expresses a version of the protein that can no longer self-interact, then we can see something very similar. This works in benthamiana, but it also works in Arabidopsis. So this seems to have a broad effect in different hosts. Now, what do we know about ORF57? Not very much yet. I can tell you that it seems to be at plasmodesmata, and you can see the co-localization with the PD marker PDLP5 here, which is quite beautiful. And we observed that in the plants that are infected by the mutant viruses, we can detect cell death that is associated to the vasculature. So possibly where the virus is replicating, actually we have cell death, and this in the end translates into a lower viral load, and possibly these very strong symptom-like changes that we can see in these plants. So this protein encoded by ORF57 seems to be suppressing cell death in the context of the infection to promote viral accumulation. And I would like to show you a second example. So this is TYLCV, and it's work by my PhD student Shuyi, who's really amazing. And she found this ORF41 in TYLCV that is predicted to interact with autophagy-related or ATG8. It interacts with this protein through this typical IME domain, so ATG interacting motif. And this is work by Tolga Bozkurt at Imperial College London. So when we have this interaction, we can compare the localization of ATG8 and ORF41, and then we can see in planta formation of these puncta that are likely autophagosomes. In this co-IP experiment, the interesting part for this purpose is this in this little red box. So we can see the interaction between ATG8 and ORF41 in the case of the wild type. But if we mutate the IME domain in these 2 residues in red, then the interaction is largely diminished. So indeed, the interaction is real and is happening through this canonical IME motif. And if we generate a mutant version of the virus that produces this version of ORF51, so the one with these 2 residues mutated, then there is a very clear penalty to the infection. So indeed, ORF41 has a proviral function, and this proviral function depends on its interaction with ATG8. So summing up what I have told you about these putative novel proteins, I have shown you that PBG is a new extracellular protein, and it acts as a PAM mimic, most likely. This ORF57 from CPCTV forms a putative channel that prevents vascular cell death during infection. All 3 Gemini viruses that we have studied so far contain at least one new protein that is predicted to interact ATG8, such as ORF41, and we have confirmed the interaction already. And splicing generates isoforms that are functional specialized, although I did not have time to tell you today, but some of you might remember Delphine's work. So basically, all in all, we think Gemini viruses encode additional proteins, but it's more than that. They encode additional functions. So we can actually learn a lot more if we just look into these newly uncovered repertoire. If you consider how the genome is organized, then you will probably realize that most likely there are also new regulatory mechanisms that have to be described. So just in case some of you think an image is worth 1,000 words, then this is where I think we stand right now in our understanding of these viruses and possibly many others. And with this, I'd just like to thank the people who did the work. I'm super lucky to have a fantastic team working together with me. I would like to thank our collaborators. I'm listing there only the ones directly related to the work I presented today, funding agencies, and of course you for your attention.
Thank you very much, Rosa. Outstanding talk, very exciting, and congratulations. And now it's time for you, for all of you. Who wants to make the first question? I'm sure today we will have many.
Thank you, Rosa, for a wonderful talk. One question, so about this, what do you think this peptide, you know, you show that when you do RNA-seq, you have more than 4,000 genes regulated. So how are you going to, I'm sure you're looking for a receptor, so who is sensing this peptide and how, and do you have any clue? No, yes?
I have a slide for you. This one. Yeah, so our first hypothesis, which is not the only possibility, but we thought the peptide might be perceived at the plasma membrane by a receptor complex, right? Not the only option. So then Laura tested the two main core receptors, so BAC1 and SOBIA1, right? So BAC1 is for both RLKs and RLPs, SOBIA1 for RLPs. And then she found both of them are actually required. So if we silence either BAC1 or SOBIA1, then we don't have relocalization anymore. And in fact, SOBIA, so that's what you have here. So this is, in this case, this is a SOBIA mutant. And then you see how C4 here at the plasma membrane, so this is the MOC condition, and here with FLAG22, it moves, right? Because SOBIA1 is not required for FLAG22 perception. But if now we treat with PVG, while we see the movement in the control, in the silence, in the mutant plants, C4 largely remains at the plasma membrane. So we think that SOBIA is required. And we currently have 2 candidate RLPs that we are testing. But it's too early to say.
Yeah, next question.
No, it's fine? Yes? Okay, so I'm missing.
Again. But just shout. Yeah. Yeah. We have tried. It's not easy because it's 5 amino acids, right? So of course you get hits. But so we have been trying to compare it to known peptides in the plant, but still, like, we have not found anything super clear. But I think this we really need to do. Well, I don't know. Probably there are some ways, but we have been doing largely by hand, and we have not seen anything obvious. However, I have to say one of the RLPs that is our top candidate, and this is based on mass spec data with the biotinylated peptide, is a receptor for different pumps. So it's one RLP that can perceive at least 3 different peptides. I don't know whether this means something or not. We still need to confirm whether this is the receptor. But yeah, so it would be great to know, but it's a little bit complicated because it's so short. And in the signal peptide, this can be anything, right? So.
Next question.
Everybody's totally unbiased.
I'm really not into lab pathogen interaction world, so
Yeah, so I mean, that is a tricky question, and I don't have a definite answer for you. My gut feeling is that it all relies on the hierarchy of expression, right? So something we found is that for PBG to be expressed, the rep protein needs to be there. So somehow rep directly or indirectly will be activating expression of this other one, right? So that already determines when, like that, you know, the temporal timeline, like when this is going to be expressed. So C4 will be expressed early, it will go to the plasma membrane, and it will start suppressing the cell-to-cell movement of RNA silencing. It will start triggering symptom development, which will take a little bit of time, I assume. But then at some point, once rep accumulates, I guess it activates the expression of PBG. Then PBG is secreted, then it triggers movement of C4. So I have to say C4 is retaining PD, so it doesn't move from PD, so it will keep acting, you know, as a suppressor of the movement of silencing. But then the rest of C4 from the rest of the membrane, it's endocytosed, right, and goes to the chloroplast and suppresses defense from there. But one thing we have to think is that the virus might keep pumping C4, right? That's not the population. There will be more coming to the membrane. Once defense goes down, there's a chance that this is kind of not needed anymore, right? And then there will be more accumulating at the plasma membrane. And at some point, this will just be shut down because then the late genes will come up. So, you know, I think it's a little bit complicated. We know we also have splicing, controlling viral life cycle progression. So I think it's probably a rather elaborate mechanism, and we are still missing quite some factors, I presume. Now with all these new open reading frames, then the hierarchy of expression is going to be much more complex than we thought. Something that maybe I should clarify, though, is that PVG does not trigger a full-blown defense response. So it does not activate MAP kinases. It does not lead to accumulation of reactive oxygen species. It does not confer protection to bacteria. So it does lead to changes in gene expression and chloroplast clustering around the nucleus. Those are the only readouts that we have found positive. Thank you. It means more without the microphone.
It just something that caught my attention, a comment you said now about C4 that is relocating from plasma membrane to chloroplast, but not from plasmodesmata. Of course, I'm curious about that. Do you know? Do you have any guess on why?
I have absolutely no idea, and probably you could make up, come up with a more plausible hypothesis than I could. So when I asked, I was told maybe it's just a physical thing, that it cannot easily be endocytosed from there. I really have no idea. I really don't know. I mean, it's.
Do you know if it is within, inside the plasmodesmata nanotube, or it is in the sides of the plasmodesmata?
I can't tell you that. I don't know. Yeah, no, I don't know.
First of all, thank you for this magnificent presentation. Related with the question of my partner, I would like to know if you have thought about the possibility that C4 is transported to the chloroplast by membrane contact sites instead of vesicular transport. It could be a very plausible possibility, as they are faster than the vesicular transport, and the effector could be faster than this.
Yeah, absolutely. I mean, you're totally right. And this was kind of our first idea. Because also, since we are imaging epidermal pavement cells, which is probably not ideal, but it's what we can do so far, and the chloroplasts are really close to the plasma membrane, right? So this was our first thought too. But then when Jihao started dissecting this, then he found these dots, right, colocalizing with endosomes, and then we found these endosomes clustering around the chloroplast. So I mean, I have to say, it's not, these possibilities are not mutually exclusive, right?
That was an excellent talk. One of the best I've been in the last 20 years. There are so many things you said that it's hard to keep track of everything. But now that you have identified all these new proteins and possibilities, I'm thinking about the evolution of virus, which opened a whole new window because, you know, now you can check, because it's a law of evolution, and there's so many replications, you can imagine always, you know, how they can evolve. So if you look for virus that you sample, probably Eduardo sampled many years ago, and you sequence that.
I don't know what he's trying to imply.
No, I mean, how can you put this into evolutionary context of virus? Because before you just have the sequence, right? But now you have the sequence, and then you have something more than the sequence. You have a lot of information of a lot of new proteins. That how can you evolve on this?
Yeah, so I guess that's the next challenge, right? So, I mean, some things I can say. So some of the new open read frames are very conserved, and some are absolutely not conserved, and you can see them only in specific clades, right? So this is one of the first criteria that we use. Also, I mean, one very good indication that we also use experimentally is that you get reversion. So when we infect with the mutants, you know, like if there is evolutionary pressure, you should find reversion. Of course, this depends on the host you're using, but we also see that. Then how everything evolves together in nature, if that's where you're aiming, honestly, I find that absolutely mind-blowing. So I can't, you know, I don't know if I will ever be able to understand this. So I mean, viruses are populations, right? So this is also something that we have to take into account, but still. Now, if you consider that that viral proteins also seem to interact with one another, right? There are even more pressures. So I honestly can't answer that. I would love to. I probably will devote the rest of my life to that question, and you know, I'm doomed.
Hi Rosa, thank you so much. Really nice talk. I have only two little questions. The first one is, how is supposed to be produced PPG? Because if I'm not, it's in the opposite direction of REP.
Mm-hmm.
And then it's produced within the viral context, but you also have a transgene, no? Only expressed in REP.
Yes.
And then it's produced also from that.
Yes, so we tested that would imply that the REP sequence upstream of the PVG start site, that should be acting as promoter, right? So this is what we tested, and indeed we found activity only if we have the REP protein. So that DNA sequence it would be, if you go in the direction of REP, it would be the end of REP, right, like the 3' of REP, but in the opposite direction, it seems to have promoter activity if you have the REP protein.
Okay, so it's related to the second question. When you look at the nanopore data, you have many transcriptional start sites. It's not only the two typical ones.
No, it's a mess.
Poly-A sites.
Yes. Yes.
Thank you.
Thank you very much, Rosa. This is my best talk in the last 30 years. Anyway, I'm going back to evolution. How do you reconcile these things? Because, I mean, taking account the mutation rate of those juvenile viruses, the fact that you have so many open frames there is, I cannot believe they are common to keep concerned. Maybe there is a core of proteins that are conserved, that they require, they're basic, but then it seems to me like an evolution experiment of the viruses producing proteins that are usable, and when they are used as selected, but they don't care really about that. What is your idea about how is the evolution working
Yeah, so we think, I mean, based on conservation, we think the new open breeding frames are younger. So they, in general, I mean, some of the new open breeding frames are actually very conserved within certain family genera, such as, for example, this V3, right? It's super conserved. But some of the others we find in really in specific clades. So we think probably they are younger. How does this evolve and is maintained? I really have no idea. So again, we are talking about populations here, and they are exploring, right? All the possibilities, and then whatever works stays, and whatever doesn't gets purged out, and then it will depend, of course, on the host and the conditions and everything. So I think things will keep appearing and disappearing, I would assume.
Completely changed the idea. That completely changed the idea that we have about virus evolution. If you think about that, I mean, we always see the picture where you see the open breeding frames of the virus has been very well established. And maybe it's not only viruses what happened there.
It's not. It's not. I mean, I think this is also, I mean, the fact that there's more, I think this also, I mean, this has also been demonstrated by now. So there's a paper, so Xueping is the last author, Xueping Zhou from China. This was published in 2024. He showed that RNA viruses also have coding capacity in the negative strand, which was supposed to be non-coding, right? And in this paper, I mean, it's a really beautiful piece of work. And this is demonstrated for one plant-infecting virus and for COVID, actually, for SARS-CoV-2. And in both cases, they showed this is produced during infection, and this has relevance. So we really have been missing out. And I mean, we are also looking at PVX just as as an unrelated RNA virus, and we find the same thing. So this is the map, this is the canonical map, and then these are new open breeding frames that we find. So except for one, all of them are in the reverse orientation. But actually, we find many of them which have clear effects on the infection. So I think this is a general, it's a general thing that we have been missing out more open breeding frames in viral genomes. I mean, we are, life is not getting any easier for any of us. But
amazing talk. I have a question regarding your ATG8 interaction with the viral protein. If I have understood correctly, you see this interaction in your hypothesis, or you see a provider effect in this interaction with the autophagy pathway. So what's your hypothesis in this provider effect? Because sometimes this is also related with the stress response, that the plant will degrade the viral. So in this case, what's the hypothesis?
so we don't know yet. We don't know yet. Actually, the infection results are really, really recent. So they are from last week. Yes, very fresh. So we don't really know yet. It could be that it's just somehow impairing degradation of viral proteins, but it could also be that it's more linked to plant defense. So there's a relatively recent preprint showing that autophagy can act as a rheostat in response to viruses to impair overactivation of defense. So maybe it's linked to that. We really don't know. Right now, all possibilities are open. I just find it very interesting that no Gemini viral protein was, as far as I can tell, was shown before with an AIM motif and binding these ATG8. And then suddenly when we start screening with the new open breeding frames, that we find one per virus, right? I mean, we have 3. Also, PVX has 1. So we are collaborating with Tolga Boskurt, and they were looking at the canonical proteins, of course, because that's what they could find, and they didn't see anything. So they were a little bit desperate. And then when they contacted us, we sent them the whole lists with everything that had been predicted. And then suddenly, some of these viruses have even 2 that interact with ATG8, and the interactions we have confirmed already. So.
Particularly amazing from the point of view that evolution is happening in medicine. But if I'm not wrong, you have mentioned a control that you have in which a single mutation in a glycine preventing installation was capable of sending a C4 to the chloroplast and then fully complementing at least on viral load. So that sounds like a much shorter way to get to a point. So obviously there's something more. So can you tell us a little bit more about what C4 is doing to the membrane?
Yeah, I mean, I think C4 is needed at the membrane for, on the one hand, this suppression of the cell-to-cell movement of silencing, and then for symptom development. There's a chance there's more, surprisingly, because what we are doing now is doing competition experiments, so mixed infections, and which is always a good idea, right? And suddenly, when we do the mixed infection, the wild type does outcompete the mutant that no longer produces symptoms, but we thought was doing nothing else because in terms of viral load was the same. Well, it's clearly not the same, right? So there's more. Why not simply have a separate protein going to chloroplasts? I mean, I think two potential reasons. One, I mean, there's really very little space, although well used. But also maybe it's about timing, right? So when do you need to be in the chloroplast? Maybe you don't need to be suppressing at all times. There's something, I don't know if I kept that. Yes, I kept that slide. So there's something that is very interesting. So this is a paper in Press, so it will be out very soon. So it's from Shueping Zhou's lab. And here they study two different isolates of the same virus found in China with two different versions of C4. So one of the viruses has a C4 that is super efficient going to the chloroplast and suppressing SA. That's this one here. And the other variant actually cannot do the job, so SA is actually quite high. Now, in this case, when SA is high, there's callus deposition in the PD and the virus stays in the phloem. While if C4 is super efficient, suppressing SA, then there's no callus deposition and the virus moves. So it escapes the phloem. Now in nature, this one, so the one with the least efficiency for is more prevalent. And this didn't make much sense until they tested transmission. And it turns out that if you escape the phloem, there is a penalty for that because the insect vector cannot find you anymore, right? So there's a trade-off. It's not, I think going all in and just killing defense is probably not a good thing either.
Thank you very much, Rosa.
Thank you. Thanks a lot.
I wanna dance by the water beneath the Mexican sky, drink some margaritas by a string of blue lights, listen to the mariachi play at midnight. Are you with me? Are you with me?